Hey everyone, so we've all seen this image a million times, right? DNA, that beautiful, twisting double helix, it's like the icon of science, but we tend to think of it as this rigid, static thing.
But here's the thing. What if that image is just a single frame in a much, much longer movie? Is this classic shape really the whole story?
No way. Not even close. See, inside our cells, DNA is alive with motion. It's constantly bending, twisting, and changing its shape. It's a shapeshifter. It has this secret life, this incredible dance that's happening all the time. And today we're going to pull back the curtain and see how scientists are able to watch this dance that's completely invisible to the naked eye.
So here's how we'll break it down. First, we'll look at DNA's different secret shapes. Then we'll tackle the massive challenge of actually trying to see them. After that, we dive into how scientists build virtual DNA, find its most stable forms, and then simulate its dance. And finally, the best part, we'll uncover what these amazing simulations are telling us.
Okay, first up, let's talk about these other shapes. Turns out that classic double helix we all know is really just one of DNA's favorite poses.
What's really wild is how much the local environment affects DNA's shape. That B DNA, the one we see in textbooks, that's what it looks like when there's plenty of water around. But if you start to dry it out, it scrunches up into a different right-handed shape called aDNA. And get this, if you crank up the salt concentration around certain DNA sequences, it can completely flip its direction and become a left-handed helix called zDNA. The same molecule, totally different structures.
Okay, so this brings up a huge, huge problem. If DNA is constantly twisting and changing like this, how on earth can we possibly study it? And these aren't just random little wiggles. These shape changes are absolutely critical for how our genes work. But they happen on a scale that's, well, it's almost impossible to comprehend. We're talking nanometers, billionths of a meter, and picoseconds, trillionths of a second. It's way too small and way, way too fast to watch with any microscope. So, how do we see the invisible?
Well, we turn to computers. Just like this quote says, bioinformatics gives us the power to build a digital twin of a DNA molecule. It lets us sidestep the incredibly difficult, sometimes impossible lab work, and instead study its every little move in a virtual world.
So yeah, if we can't watch the real thing, let's just build a perfect virtual copy and watch that instead. Let's get into how scientists actually create this digital twin of DNA.
It all starts with a known DNA structure, something you can just download from a scientific database. Then, you basically give the computer a map, defining every single atom and all the chemical bonds connecting them. And then comes the magic step. You apply what's called a force field. After that, you just hit go and let the supercomputer do its thing, calculating how the whole molecule is going to behave.
So what on earth is a force field? It's not science fiction, I promise. You can think of it as the physics engine for our virtual molecule. It's a really sophisticated set of equations that spells out all the rules of reality, how atoms attract each other, how they repel each other, how their bonds can stretch and bend and twist. It's basically the rulebook that tells the digital DNA how to behave like real DNA. Okay, so once we have our virtual DNA running on its physics engine, there are two main things we can do. There's a technique called energy minimization, which is basically like taking a single perfect photograph to find the molecule's most relaxed and stable pose. And then there's molecular dynamics, which is like hitting the play button and watching a full movie of how the molecule actually wiggles and jiggles and dances over time. Let's dig into that first one, the molecular snapshot. This is all about finding the most stable possible form of the molecule through something called energy minimization. The best way to think about this is to imagine a really hilly landscape. If you put a ball anywhere on those hills, it's going to roll down until it finds the very lowest valley, right? That's its most stable spot. Well, energy minimization algorithms do exactly that, but for our DNA molecule. They nudge every single atom around, searching for that one single arrangement, that one pose that has the lowest possible energy. That's its most likely shape. But a photo is just one moment in time. What we really want is the movie. And for that, we need the incredibly powerful technique of molecular dynamics. It's time to press play. Right, this is where it gets really cool. Molecular dynamics, or MD for short, doesn't just look for a resting state. It actually gives the atoms kinetic energy, which is how you simulate temperature. And then it applies Newton's classic laws of motion to every single atom. The computer calculates the forces, figures out where each atom will move in the next tiny fraction of a second, and then does that again and again, millions of times. The result is a trajectory, A full-blown movie of the molecules dance. Okay, so we built the virtual DNA. We made a movie of it. What's the payoff? What have these simulations actually revealed about the secrets of this dance that we didn't know before? Oh, a ton. For instance, one of the most basic movements in DNA is something called the repuckering of the sugar rings in its backbone. It's this tiny fundamental little flip. Well, these simulations showed that this happens spontaneously all the time, and it happens in just a few picoseconds. That's a few trillionths of a second. It's mind-bogglingly fast. And it doesn't stop there. Using these methods, scientists have been able to map out the exact step-by-step pathway for how DNA flips from the B form to the Z form. They figured out that certain sugars in the backbone are way more flexible than others. They've watched DNA strands bend back and forth on their own every 26 picoseconds. They've even been able to calculate the absolute perfect, most stable twist for a DNA helix. It's 10.5 base pairs for every full turn. And this is the really critical takeaway. These computer simulations aren't just for confirming stuff we already kind of suspect from lab work. They're true engines of discovery. They let us see brand new things about the blueprint of life that were totally impossible to observe any other way.
Now, this is all fascinating stuff, for sure. But, why does understanding this incredibly detailed molecular dance actually matter in the real world?
Well, it comes down to one of the most fundamental rules in all of biology. Shape determines function. A molecule's 3D structure dictates what it does. So by understanding exactly how DNA twists and bends and breeds, we get a much, much deeper insight into how life itself works, from how our genes get switched on and off to how vital proteins know exactly where to bind to our DNA.
And that leaves us with a really powerful, forward-looking question. If we can build these models so well that we can actually predict the intricate steps of DNA's dance, what comes next? Does it mean we can start to design new drugs, new therapies that can actually change the steps of the dance? To maybe correct a wrong move, or even write a whole new piece of choreography in our fight against disease? The dance, it seems, is only just beginning.